So this is also a bispecific antibody, as you said, developed by Pfizer, and it's one of several bispecifics that is being presented at ASH this year. This year seems to be a year of bispecifics, with all very promising data. This particular bispecific, as you said, is BCMA directed. B-cell maturation antigen is an antigen highly expressed on myeloma cells, as well as CD3 directed, so CD3 expressed on T cells. It's on an IgG2A backbone, so it's based off of antibody technology. And the current study that's being presented at ASH on Monday in poster form is evaluating subcutaneous dosing administration in a dose-escalation fashion. The compound has previously been evaluated in a dose-escalation study in an IV formulation, and preliminary efficacy was observed. The idea here is to see if subcutaneous dosing can lead to a wider therapeutic window. 30 patients were enrolled at the time of study cut on this trial, up to a dose of 1,000 micrograms per kilogram. Of the 20 patients treated in the highest dose levels, the response rate was 80%. Importantly, this was a heavily pretreated patient population with approximately a median of 8 prior lines of therapy, and almost a quarter of the patients had prior BCMA-directed therapy exposure, including prior CAR T-cell therapies, as well as BCMA antibody drug conjugate-based therapies prior to enrollment on this study. Of these patients, there were 4, 3 responded. From a side effect perspective, the most common observed adverse event was CRS, which occurred in 73% of the patients. This was generally grade 1 or grade 2 and occurred within 2 days of administration. 40% of the patients received tocilizumab. I believe 18% had dexamethasone. So overall, a very promising antibody, bispecific antibody drug, with encouraging signs of efficacy at its therapeutic doses. And this compound will go forward in additional trials as this study develops in further evaluation of its single agent efficacy as well as in combinations.